Okay, good afternoon, everyone. Welcome to Willem Nielsen's talk today. Willem will be speaking to us about computational foundations of biology in the Wolfram Physics Project. Willem studied computer science originally, and he was a student at the summer school last year, where he was interested in complex systems, among other things. Along the way, he got more and more interested in the Wolfram language, and he somehow got really keen into computational biology. And since then, he's joined the Wolfram Institute, where he's now a full-time researcher working on computational evolution, computational medicine, and all things related to computational biology in the WPP, meaning that the rewriting cellular automata slash hypergraph rewriting approach to computationalizing biology. So today we'll be hearing about some of the lovely work that William has been doing at the Institute. Thanks, William, for being here, and take it off.
Yeah, thanks, Xerxes. I'm excited to be here. Yeah, so like Xerxes says, I'm going to be talking
Your audio is a bit down. Maybe you could adjust stuff or come closer or speak louder. Either option could work.
Yeah, let me try this.
Is that better?
Yeah, some.
Okay. Yeah, so I'm going to be talking, as Xerxes mentioned, about using hypergraph rewriting models to look, in this case, at morphogenesis. So morphogenesis is just the process that biological systems go through to when they start from some small for in the case of animals, it's like you start from some embryo, and then it's the process of transformation that gives the system its shape and size. And so I think hypergraphs have some potential to model that process, and so that's what I'll be talking about today. So just to give you some background, Stephen Wolfram looked extensively at what's called developmental biology, which just means kind of how organisms develop, like from in the case of animals, as I said, from embryo to full growth state. But he looked a lot at, for example, shell. So this is just showing a model of a simple model of shell growth and how different parameters can kind of affect the shape of the shell, and how the variation of the parameters in this model looks very similar to how actual shells look in biology, how actual shells vary, I should say, in biology. And so he also looked at plant growth or leaf shape, and using like a simple branching substitution system, was able again to approximate a lot of the variation that we see in leaf shapes in biology. So one of the things that's in common though about both of these systems is that they have a fixed growth area, meaning so in a plant only the edge of the stems can grow, and then the inside piece of the organism is fixed. Same with the shell, like only the outer edge can grow, and then the inside becomes fixed, which makes it a little the possible shapes kind of a little bit simpler. So the hypergraphs, I think, have the potential to kind of fill that gap, because they're more flexible in how I should say fill the gap of animal growth, which is that it's more plastic. So like, when animals grow, they don't just grow from the edge, they can grow from any piece. And so because of that, the potential shapes is more complex. And I think hypergraphs, because the way in which the rewriting system works, has the potential to fill that gap of modeling a higher level, more complex organism growth. So just to give you a little background, I know you guys are or at least some of you are familiar, but the way that hypergraph rewriting works is you start with some graph. So in this case, we start with this graph that has two kind of edges pointing to itself, with a node with two edges pointing to itself, I should say. And so then we apply this rule. So in this case, it's this rule. And so on the left side of the rule is like the kind of matching graph. So it's looking for any instances within the graph of this particular edge and node pattern. And so then if it finds it, then it replaces it with the graph on the right-hand side. So in the Wolfram Physics like technical introduction, they do like a really in-depth explanation of this of how hypergraphs work. And if you're interested, or I'm sure you guys know about it, but I encourage you to go check it out. I'm not going to talk too much about it just because it's kind of complicated, and I'm trying to focus on the biology. But in this case, we have, so like for example, this first case, we have again, like I said, two edges pointing to the same node. So that is seen, it's a little bit confusing, but it's seen as this kind of like this pattern, but it's just all with the same node. And so then what the dashed lines mean that it matched on the pattern. And so then in the next one, the red edges are ones that were added. And then the dashed ones are ones that are gonna get matched and replaced for the next one. So we're just kind of doing this replacement process, and we can think about it in biology terms or morphogenesis terms as each node is like a cell, and then the edges are kind of like the state of the cell. So if the node is kind of in a state where it's going to get matched, where it has like a chain, then it's kind of like a growth state because it's likely to get replaced and kind of divide, so to speak, and increase the number of nodes. And then if it's in like a non-chain state where it's not going to get matched, then it's kind of like a quiescent or a cell that's not going to divide. So it's kind of has a effect of limiting the growth. And so what that means is you can kind of model kind of the important thing, or kind of the key puzzle of morphogenesis is how do organisms grow and then stop growing. And so I kind of wanted to ask, does anyone have, because at first when people hear this, it's a little confusing. So I think it would be a good time. Does anyone have any idea about how this would work, about how organisms grow and then stop growing?
I have no idea. All I know is I think Stephen had mentioned this a number of months ago. And at first I had come across it. It was presented more of like almost a woo-woo type of thing. And then I found out that John von Neumann apparently had been working on it. And I'm like, okay, that probably is something there. But it was interesting that you guys actually found that like this did model like different behavior. Am I wrong?
Well, so we'll talk about it more, but I do think the hypergraph and also cellular automata and string rewriting can indeed model the process of morphogenesis and how things grow and then stop growing. But I don't know, anyone else have any other idea?
It's interesting because, as far as I understand, some organisms kind of don't stop growing. They're kind of only limited in their growth by environmental factors like how much food they have and how easy it is for them to be preyed upon by other organisms. So I'm trying to think in my head like how you would kind of bake in a stopping mechanism where they reach a certain phase of their life cycle. I know, for example, in eels, there's this idea that a rare genetic abnormality can occur where they early on in their life cycle, they go through a stage of really rapid growth, and that stops, and they kind of transition into a different stage of the life cycle. But there's a rare genetic abnormality that can occur where they don't hit that switch, and so they get stuck in that early life cycle stage and just grow really big.
Mm-hmm.
I mean, we may not need to even bake in a stopping condition. It could just be that the computation stops or halts.
Yeah.
So that could be like a death or something like this, I suppose.
Like if it's a rule that like limits the number of edges, like an edge limiting rule.
Yeah, so you guys are definitely getting closer. So, or kind of closer to what I'm imagining it as. So in this particular rule, this is actually a halting rule or a halting hypergraph. And so the way it works is if you look at the left-hand side of the rule, it's kind of like matching on chains, but then what it replaces it with, there's no chains in kind of the right-hand side. So eventually you basically just run out of chains to replace, and then there's no more no more growth can happen. And so it's kind of like I think it's like it's a good way to think about how morphogenesis would work, because it's like a negative feedback loop, where like kind of like you're stretching a rubber band. As you stretch it more, it becomes harder and harder to stretch it. And so then you can kind of land at some final state or some shape that you would, the organism would like to be at, so to speak. But yeah, so this is kind of like a minimal case where the program halts. But obviously in biology, we have much more complex forms. And so the question is, can we kind of model those? And before I get to that, though, I should give you just a little more background on the hypergraph, which is that in this particular halting graph, we had a very specific order of choosing our update rules. But if we choose random ones, then what's interesting is we actually get different shapes. So kind of akin to, I think about it as how, small changes to a biological system, whether that's the environment or the genetic, perturbations, can lead to changes in the final shape. But what's interesting also is that no matter which order you choose, it ends up terminating. So again, similar to biology, how it's able to handle perturbations and still end up at least a similar shape, right? Okay, so again, going back to can we model more complex kind of shapes? And so these are some examples of rules, more complex rules that are able to produce more complex final shapes. And so what's interesting about these is like it's really unclear as to, like in the previous case, we could kind of analyze it a bit and see why it terminates. But in this case, it's much harder to tell, why does it terminate at this particular shape at this particular step. And so kind of looking more into that, we can see this is like showing each kind of sample steps for the rewrite. So like this is the first 3 steps of rewriting, and then this is like the last 3 steps down here. And again, it's very unclear and complicated as to why, at step 241, the system stops growing. And so it's kind of like, I think about it as akin to the complexity we see in biological systems. And even though, they can be robust, the mechanism by which they, achieve their goal, in this case of terminating at some, large vertex count, is very complex and hard to study. And I think it's part of the reason why biology is so, difficult in such a large and complex field. Okay, so we can also look at different rewriting orders for this more complex hypergraph. And again, what we see is that there's a lot of variation in the number of steps. I should say the number of steps that it terminates at. So that's what this number means. And you can also see different, shapes of the final state from different rewriting orders. And again, I think about it as like, small changes to the environment or genes can lead to different shapes. So kind of going along with that, we can look at the growth curve of this particular hypergraph. So that just means at each generation, how many vertexes are in the graph. And what's nice is, similar to kind of the growth in biological systems, we kind of see this increase in speed of growth in the beginning, and then we see it taper off before it reaches its final state. So kind of how like adults slow down their growth. And we can look at the same graph, but with multiple different rewriting orders. And what we see is, again, there's so a lot of them terminate within kind of the 250 to 20 step range. But there is definitely some variation within that kind of, you could potentially think about that as like how, there's variation in the mass of humans, for example. And what's cool is, I should say before that too, that this is kind of a similar idea, but just showing the distribution at each step among different rewriting orders. So it's kind of a similar idea, but we again see the kind of smooth variation, but there is kind of like a mean shape, so to speak. Okay, so what I was going to say is what's cool is we can look at the Wolfram language has built-in human growth data, and we can compare our hypergraph growth with how humans grow, and it's kind of nice that the growth increases in speed in the beginning and then tapers off just like our hypergraphs. And again, there's kind of large variation in mass between humans. And so this isn't like this one kind of looks a little different. This one doesn't have as much kind of a curve, but we can look at other hypergraphs that do fit better with the human case. So that's like this hypergraph, for example, which is kind of cool. And then kind of zooming out, we can look at the space of all of growth distributions in a bunch of different hypergraphs that halt. And so, yeah, that's what this is showing. And the most interesting one is this first one here, which is interesting because it's showing there's kind of a bifurcation between two different halting states in the hypergraph, depending on the rewriting order. And if you're familiar, it's similar to the concept of like a Waddington landscape, where there's kind of, depending on the conditions or environmental genetic, the organism can kind of get pushed into one of two phenotypes, but then within those phenotypes, there's variation among them. Sorry, let me get some more. Okay. Yeah, so I think this is also like a rich area for further study, and kind of the area of distributions is complex and I think needs to be understood better. Okay, so but how did I get to these complex hypergraphs? So the way I did it is kind of inspired by Stephen Wolfram's recent work in his model for biological evolution based on cellular automata. And it's a similar idea in that we start with just some basic kind of rewriting rule, and then or the algorithm just changes randomly one edge at a time. And so that's each rule here. I should say on either side, it can change one edge at a time. And so each rule here is kind of showing successive mutation steps in that process. And so then we we keep the changes to the rule that lead to a terminating length with a larger vertex count. So you're kind of just looking for larger and larger quote unquote organisms. And yeah, the fact that is able to lead to, or the fact that kind of emulating biology, we can do this kind of evolutionary process to find rules that develop into larger and larger, more complex shapes is interesting. And it's a nice way to model biological systems because that's how they arrive at their shape. And so I think a lot of the complexity that we see in biological systems comes from the fact that they are evolved in this way and not like engineered, for example. And so like if we were to kind of engineer a hypergraph that terminates at, vertex count 1,000, for example, I think it would look very different and not biologic. Yeah. So one of the kind of mantras of Stephen Wolfram is really focusing on the simplicity of models and trying to keep them as simple as possible. And so one way we can do that in this case is to look at the case of string rewriting as opposed to hypergraphs, which is a similar process, except that, and I'll explain this in a second, but there's just, instead of replacing a graph, we're just replacing substrings within a string. And these are examples of kind of string rewriting systems that terminate at some, large step kind of. And again, what's interesting is the complexity, these kind of look biological. So just to explain a little bit more about string rewriting. So the idea is you just have a string here. So in this case, A corresponds to this red color. And then we just replace at each step. So, these are the rules. So this first rule says that if you see a red block, then you replace it with a blue and a green block. And so that's this next step, and you kind of recurse on that process. And in this case, we're looking for string rewriting systems that terminate. So this one, at whatever step this is, it leads to itself. And so we can think about that as kind of the organism, quote unquote, like not growing anymore. Okay, so what about more complex string rewriting systems? So one of the things we can do that's kind of interesting is...
Sorry, is it okay to interrupt really briefly?
Yeah, go ahead.
If you can go back to the previous one and just look at that first step, the second rewriting step, can you just go over that really briefly and explain the beige pointy thingies and why the blue starts making this funny shape?
Yeah, okay, so so one thing to note here is just like with hypergraphs, the order that we choose to rewrite in matters. And, we're kind of just picking an arbitrary one in this case. But so again, like I said, we take the red block, we replace it. And then, so in the second step, because of the ordering function that we use, we end up replacing the green block with a yellow and purple block. And the reason why this one looks like that is because, well, there's no replacement rule for the purple, so it just leads to itself. And so any area of the string, I'm not sure if this is answering your question, but any area of the string that doesn't get replaced kind of just stays the same. So that's like the reason for the vertical block there. Does that make sense?
Yeah, okay. So the vertical block, so where the beige doesn't apply, is there is no rule. So it's not just that it does not change, but there is no rule specifically.
Yeah.
Okay, thanks.
Is it that there's no rule or there's no rewrites available?
No, it's just that in our particular string rewriting rule, when we see a purple one, we just ignore it.
Okay.
Okay. So so we can kind of approximate more complex string rewriting or biological systems using string rewriting systems in this case. So I just used the fitness function using a similar adaptive process to the hypergraph that uses a fitness function to match the growth curve of a human, for example, as kind of like a toy example. And so what's interesting is the types of rewrite systems, again, that we get when we do this is we kind of get these complex forms, but there is some variation between them. So it's kind of like a smooth variation like the one we saw with the human growth. So so this is that particular rule kind of fit to the human growth curve. And, it's not a perfect fit, but it's kind of shows you the kind of the type of system that you get when you're trying to, fit to some shape. So that's the end. So thank you to Dugan, Xerxes, and Stephen for ideas and help on this. And if you want to see the code, it's here. I don't know, you guys can take a picture, I guess. But yeah, I think it was really interesting to do this, and I hope you found it as interesting as I did. And thank you.
Thank you, Willem. That was really interesting. Can I ask a question?
Sure.
So, okay, I think toward the beginning we mentioned that in like a real organism, its growth curve is going to be a function of environment and genetic factors. With the rule in the graph or the string rewriting, it doesn't really matter which we're talking about, I don't think. Are we saying that rule is kind of a representation of both the genetic and the environmental factors? I suppose there's no way to parse out the two.
Yeah, so I think that's a good question. So we maybe just to go back to so because it's kind of like a minimal model, the way I think about different rewriting orders is kind of like small changes to the system. And you can think about it in other ways, like small changes to the rule or small changes to the initial conditions. But the types of change, like you said, because it's kind of a minimal model, we kind of take all the possible changes that can happen and just group them into this one class, which is changes to the rewriting order. So obviously, there's usefulness in like, making a more realistic model. But if we're just trying to capture kind of the essence of a very broad phenomenon like, biological growth, I think it's good to keep it simple and just use kind of this meta idea.
Yeah, I guess I'm just trying to imagine, like, if you were to take it to the next step, like how would you parse out the two? I guess if you had two sets of rules, and one rule is kind of the genetic rule, and one rule is kind of the environmental rule. I guess that would work more easily in the string rewriting system, where you do have like a set of rules depending on like what the last square was, or what color it was, or whatever. But in the hypergraph version, I'm struggling to imagine. I mean, you can have more than one rule, I guess.
Not sure about that last part, but I would say, I mean, just thinking about it now, I haven't thought much about it, but I would say that, like, to me, it seems like the kind of the underlying dynamics of the system are more driven by the rule. So that would seem more like changing the genome. And then the initial conditions, or changing kind of like a single cell, or, changing one edge at one step in the hypergraph evolution, to me seems more like an environmental change.
Yeah, okay. Yeah, I guess it would be interesting if you could model it in take three different cases, like one organism that has really great environment that it can thrive in, but its genetics are really preventing it from growing, and then inversely an organism where it's genetically it's going to really grow, but its environment is a really limiting factor, and how you could model those two different cases, or another case where it has both the genetics and the environment going for it, and it can just really grow. And if you could compare those three cases and see, what the final state, like what the final level of growth is. I mean, in the latter case, you'd expect it to have the most growth, and so on. That would be really interesting to model, I think, the kind of trade-off between environments and genetics on growth.
Yeah, I agree. It's kind of like the relationship between, in our model, like the initial conditions and the rule, and how those things vary when you change those two things.
Okay, thanks.
Yeah.
I think Matt has a question.
Thanks for a great talk, Willem. I remember your video going up onto the Wolfram Institute. I'm like, all right, I think I started to watch it. I'm like, all right, I need to have time to actually watch it rather than just listen, just because of all the visuals and everything. But could you go ahead a couple of slides to some of the, I think it was two slides forward. No, one more, sorry. Doesn't keep going, sorry. There, okay. Sorry, I couldn't help but kind of like jiving off of what Flo was just pointing out there. That first one, time generations, and that bifurcation there, for some reason my mind went to like the development of frogs early on. And I believe I'm trying to find like validated articles and everything right now, but there was some effect with, depending on the temperature, there would actually be like a difference in selection effects for the basically they might tend more towards male, they might tend more towards female, and apparently it's highly studied. I don't know a thing about it, but I just know it might be a thing.
Yeah, I think that's really interesting. I actually like this slide was one of the last ones that I did, and so I found this to be extremely interesting, but I just didn't have time to explore it more. And I was also thinking about how, something like sex, where the differentiation, creates this kind of two possible phenotypes. And I suspect that this is kind of like a kind of minimal case of how that works in a biological system. So yeah, I think it's really interesting to look into further. And that's kind of, I try to, when I do the code for these, I try to make it so that people can look at it and understand it as much as possible, so that, you know, people like you can like take it and, yeah, do more.
Who knows? Maybe it's frogs all the way down.
I have a question about the whole idea of it, like capturing the essence of it all. I don't know, like a decade or so ago, I took a class on dynamical systems, and I believe, I don't know, somebody famous, Turing or so, did a model for like why stripes evolve on animals, and like he could do that by like having embryos and then showing that just like some limiting conditions would then like lead to stripes or something. But that was actually, I think, if I remember correctly, shown to not be true in actual reality. Like that is not how it works. Like you can mathematically make it look like it is that way and like make it match up, but like the biological system itself actually worked very differently. So it was more like a coincidental matching up. And I think that's what I'm wondering here the whole time. That by what degree is this just like, you know, accidental patterns? You know, I mean, they're pretty simple patterns like growth curves or whatever. And like, at what point do we know that we actually have something of essence here?
Yeah, I mean, I think this is kind of the first step towards that. Kind of Wolfram's way of doing science, and I kind of inherited this, was just looking for qualitative similarity between the real case and the model case. And because he doesn't like really go deep into trying to like, you know, make this model exactly match, you know, the actual reality, he's able, I think, to like, you know, span a bunch of different fields and have a lot of breadth in his work. But I agree that I think there's more work if you really want to, you know, be sure that, or yeah, kind of solidify the idea, is to, you know, study it more basically, and just understand, like, for example, in this case, understand exactly why it's bifurcating, understand exactly why it's bifurcating in biological systems. And that takes a lot of time and is challenging because biology is really complicated and hard to get the right data set to kind of, you know, prove that your model is right. But I agree that it's like something that should be attempted.
But have you, I mean, is there any idea how you might be able to connect it back to, I don't know, actual biology? Like, is it like a simple test case and like super simple organisms that you guys are thinking about of seeing if there's more than just a qualitative match?
Yeah, so with the hypergraph case, I haven't thought much about how, like, the precise correspondence. But for example, we've been looking at, you know, cellular automata. I've explored much more. And so I've looked at like and this is kind of not so much developmental biology, but evolutionary, is like, the fitness landscape of our cellular automata, and how they kind of evolve to longer halting lengths. How similar is that to the fitness landscape of something like E. coli? And you know, how rugged is the landscape? Like, how many peaks are there? How many yeah, like, how hard is it to move around? Yeah, so I've looked at like specific kind of effects there, but yeah, there's more work to be done for the hypergraph case.
And what was the verdict with the E. coli?
Yeah, so again, based on like the things I've looked at, the kind of the qualitative nature of the fitness landscape is very similar. And yeah, like the number of peaks, you get similar numbers, but yeah, it's still, again, like it's kind of a lot of effort and time to make them match precisely, I would say.
I had another thought. So you mentioned about using or thinking about the initial conditions as kind of the environmental part of it, right?
Mm-hmm.
But the environment can be dynamical. So I guess if you treat the initial conditions of the environment, you're stating that you're kind of implying the environment is fixed and that's not changing over the course of the organism's growth. but the environment can change. Like the amount of food can become more scarce or more plentiful, and that can impact growth. So I guess, yeah, that's an assumption of assuming that the initial conditions sort of represent the environment, is that they're static, they don't change.
Yeah. I think that's one of those things where it's kind of like modeling a dynamic initial condition would make the model more complex, and it would be interesting, but yeah, it's kind of like not the Wolfram style, really.
Yeah.
I think the general idea is, because Stephen's blog post is called a minimal model there, where I know Willem was talking about like a fitness condition there, and you can have a very complicated rule, but ultimately living creatures are very complex. That's why I'm like trying to figure out the fabric of reality actually seemed easier.
Yeah.
Humans are too complicated, in my opinion.
Yeah. I mean, like I said, I think connecting this, I've kind of like done more work here on the model side, understanding what's going on, and just kind of finding potential candidates for, hypergraphs that could be a meta model for biological growth and morphogenesis, but connecting it to actual cases is difficult. Like I looked at trying to find simpler cases than humans, like the growth of worms, for example. But the data in that case so far, I found it to be like really sparse and kind of not good enough. But yeah,
haven't we fully sequenced like the nematode or so, a very small organism where it only has so many? But I thought we like managed to like sequence quote unquote everything.
Well, there's a difference between sequencing the genome and then looking for what we really want in this case is understanding at time steps from the time of seed to full growth, all the kind of shapes and the state of the organism at every step along the way, and then also that with a bunch of different environmental and genetic conditions, so it's kind of complicated, but I'm sure that there's better data sets out there than the ones I found. We've just got to find them.
The nematode did have its connectome, its brain mapped out and all its connections as like one of the first connectomes that was done, because it only had like 100 to 200 neurons.
Okay. So listen to what Dugan
Which is
said
wild,
my
because
heresy.
I mean, it's an organism doing things like moving around, looking at things, reacting, finding food, finding mates, doing all that on 200 neurons is, crazy.
Those are the C. elegans, right?
Yeah,
Yeah, it's the C. elegans. They have about 312 neurons, actually, more or less.
Okay, that's more reasonable.
And that's the one that's been mapped out. And I think more recently, they have also succeeded in mapping out the connectome or the neural network of the fly brain,
right.
the Drosophila, the fruit fly. So those two are mapped out. The rest are much more difficult.
How much compute did that require?
I mean, the mapping out is not. It's not so much about compute. It's actually a very, very delicate bunch of techniques, which are part of them are experimental and part of them are neuroinformatic and storing things. It's not like they are computing it. They are literally mapping out the actual connections from imaging and that kind of data.
Okay, so massive amount of brain power then. Okay.
Well, it's like fMRI imaging or whatever it is. They had to.
Well, I mean, in C. elegans, it's not exactly fMRI, but because fMRI would still be if you go monkeys and up, mammals and above. But if you're talking about below mammals, then for example, there is calcium imaging. Then there are some fiber optic techniques. You're using literally microscopic techniques to trace out the neural pathways. And in higher animals, as Dugan is saying, for humans or for monkeys, there you could use fMRI, diffusion scanning, and so on. So even the human connectome is mapped out, so to say, but not neuron to neuron wise. They've only mapped out the kind of fibers. You can actually have a look at these data sources. Many of them are open source. You just need to understand how the data is organized, and a good project would be to do some visualizations of the human connectome or of the C. elegans connectome or something like that.
I think Stephen interviewed somebody that had actually worked on, I think, the fly brain mapping.
Yeah, so I've been interfacing with a fellow from Florida who works on that model computationally. So it's like all the connectomes stored digitally, and then you can simulate it in a computer to like say, okay, well, if I simulate this type of thing, like what happened? And then for the nematodes, I think there are simulations where you can say like you tell it to like look for food in simulation. I don't know.
So we can simulate like a fly?
The brain activity of a fly, I guess.
I mean, you can even simulate the brain activity of humans just with a coarse-grained data set. On top of the actual biological data, you're using models of what kind of dynamics those neurons perform. Compared to neural networks, it's a little bit more complicated. And then you can actually run simulation models. Those are the ones that do require heavy computational power. But you can do that and try to understand all the pattern of activity for various in the way these animals or these subjects interface with various things in the outside world.
No, I was just thinking we've mapped a fly, and then you could actually like simulate a fly, its reaction to its environment going around, because we only have so much data for that. It basically, if you could create a simulation where a fly is doing its standard fly thing, that's something that everybody kind of gets. So it could kind of almost be a fly test for a level of accuracy for something that we might know. I don't know, just crazy idea.
Anyway, let's more questions for Willem. James is sharing some links over here. You want to say what those are, James?
Yeah, this is the podcast that was mentioned with Terry Sinofsky, talking about the mapping of fly brains.
Yeah, what if we had smaller brains,
So for Willem, I'm still thinking about this. Sorry. I was thinking, how could you model like a dynamical initial condition or a dynamical environment, but in a Wolfram way? And I think there are cellular automata models of like predator-prey simulations where the environment can be dynamic. And I guess in a simple case, you're only worried about one organism. You'd have one predator, if you like, in that model. But that's a kind of approach that you could do to model a dynamical environment, because the kind of prey or other squares on the grid that represent food or whatever available resources for the thing to grow, that could be dynamical in a cellular automata predator-prey simulation.
Yeah, I mean, I think the kind of using cellular automata more like spatially, I think that's kind of similar to what you're describing. Yeah, I haven't looked much at that. We mostly do like one-dimensional CAs, but I think it's interesting. But yeah, I don't have much to say on that. I mean, yeah, just one note real quick with regards to the connectome, the nematode. I was also thinking of I feel like hypergraphs, in their a rewriting system could be a good minimal model for how brain growth happens, because it is like a graph. Yeah, it would be interesting to see what fitness functions would kind of lead to more of like the branching type structure that you see in something like a brain. And I know that there's effects where you can, like, for example, look at the scaling laws of, you know, the amount of gray matter to white matter as you increase the number of size, as you increase the size of the brain. So, you know, for example, how looking at like the scaling laws of edges to nodes, and can you make the kind of make those two things correspond with hypergraph and real brains? I think that would be interesting if you know, people are interested in that.
All right, then let's thank Willem for a lovely exposition on computational biology at the Wolfram Institute. Thanks, Willem, and thanks everyone for your questions and interactions.